Hello and welcome to the 2022 INIM Conference. We want to thank you for joining us. Today we're going to hear from the Institute's clinicians and researchers as they discuss research and the various clinical approaches to ME/CFS and long COVID. My name is Sarah Moinian and I am the social media program coordinator at the Institute for Neuro-Immune Medicine here at Nova Southeastern University. I am joined by Irina Rozenfeld, board certified nurse practitioner at the INIM Clinic, assistant professor at the Dr. Kiran C. Patel College of Osteopathic Medicine, and adjunct faculty at the Ron and Kathy ASAP College of Nursing at Nova Southeastern University, who will be moderating today's Q&A session. We have an exciting lineup today of speakers. I also want to note that there's going to be a 15-minute break scheduled at 12:10. Also, if you would notice within your control panel, there's a Q&A tab. If you have any questions during the presentations, feel free to type it within that section. We will have like I mentioned earlier, we'll have a live Q&A portion at the end of this webinar, and our presenters will do their best in answering your questions. And as a reminder, this conference is being recorded and will be available on the Institute's YouTube channel within the coming days. I want to thank everyone again for joining us today. And I'll be introducing the director of the Institute for Neuro-Immune Medicine and our first speaker, Dr. Nancy Klimas. Dr. Klimas is the Dean of Research and Professor of Medicine at the Dr. Kiran C. Patel College of Osteopathic Medicine and Chair of the Department of Clinical Immunology at Nova Southeastern University. In partnership with the Miami Veterans Affairs Medical Center's Gulf War Illness Research Program, the INIM is a multidisciplinary research and clinical institute that takes a system biology approach to understanding complex medical illness, such as ME/CFS and Gulf War illness. Dr. Klimas is Professor Emerita at the University of Miami's Miller School of Medicine, a diplomat of the American Board of Internal Medicine, a diplomat in Diagnostic Laboratory Immunology, and Director of Clinical Immunology Research at Miami Veterans Affairs Medical Center. She has achieved national and international recognition for her research and clinical efforts in multi-system disorders, including ME/CFS and Gulf War illness. She is the past president of the International Association for CFS and ME and past member of the Health and Human Services CFS Advisory Committee. So I'll now hand it over to you, Dr. Klimas.
Hi, everybody. I'm so excited that you were willing to join us this morning. I know that we're a month late for the ME/CFS Awareness Day, which is when we usually do this conference, but we were putting a center grant in last month and we were at a full out run for the finish line. So we decided to do a better job by not being so rushed and putting ourselves off a bit. So I hope you don't mind that we're celebrating ME/CFS Awareness Month in June, but hey, there you go. Today, I'm really excited. I get to highlight some amazing people in our institute. I often find that I get to do a lot of speaking, and today you're going to see that we have amazing people here that are also going to be sharing their knowledge with you. And I'm very, very proud of what we've built here at Nova Southeastern University. So I'm just going to take a moment to brag because I'm allowed to. This is our pre-pandemic Christmas party that we had. There are a lot of new faces since here. We haven't been able to gather, but we're finally given permission. But you can see we're a very large group. In fact, we're a really large group, and we are fully dedicated to ME/CFS, Gulf War illness, and long COVID, complex illnesses that follow if infections or toxic injuries. That's our space, and we do clinical care. We do research from the beginning to the end. We have a translational research program where we do bench work, animal work, computational work, and clinical trials. So we're a group that is large. Most of you that know us through our clinical care team. You can see we have a nice sized team here. We're growing all the time, but and we have a lot of different types of expertise. So you'll hear today from Dr. Fornos, the newest member of our, not the newest, sort of newest person in our group, who's a functional medicine doctor, telling you what we do through the eyes of functional medicine. I thought you'd find that interesting. We're actually an interdisciplinary group. I'm an immunologist. Dr. Rey is an environmental medicine expert, among other things. We have integrative medicine, functional medicine, and Dr. Hunko is a oriental medicine expert and acupuncturist, among other things. So we have a lot of skills that we bring to the table. We do animal work, we do basic science bench work. We have a cool laboratory that's able to take new biomarkers forward into clinical use because we're a clinical lab as well. And of course, we do systems biology as one of the key features of the way we do our work. We do a lot of Gulf War illness research, and I did not include that on this slide. This is our ME/CFS research, which is certainly where we started from and where we continue to be, and we're busy. We don't have, you know, campaigns and a huge presence in the social media that we would like to be doing that even better. We do have a presence. I shouldn't say we don't, we do. But we're so focused on all this work that we're doing that sometimes I think we don't blow our own horn enough, because we do a lot of work and we move it forward. And so you'll see a lot of this work is basic science, then translational science, preclinical animal work, and then we move it into human trials. And the things in bold are human studies that are clinical trials. We've got a few others in here that are done, that aren't recruiting. So I bold the things that are either going on or about to kick off. So I know that's one of the main reasons I'm speaking today, is to tell you what we're recruiting. So one of our tricks to the trade of ME/CFS research is, you know, until long COVID anyway, and still frankly with long COVID, we are underfunded. We don't have a great big source to do particularly clinical trials work, but also some other stuff. So we've tagged a lot of stuff onto the back of other things so that we can find sources of funding, but also because it's fair to compare ME/CFS to other long-term chronic illnesses. One of them is Gulf War illness, an illness that is caused by toxic brain injury during the first Gulf War, and since, frankly, many other toxic injuries that veterans suffer. And the ME/CFS comparator group has allowed us to fund really excellent science. And this is one of those studies. Dr. Chatterjee, who's a microbiome and the brain expert, is doing a study where we've included ME/CFS as a comparator group. And we're recruiting that right now. For those of you who live in Florida, there's a slide that'll tell you how to do that. And we would really appreciate your help. Same thing as we need healthy controls, and there's no one better to recruit healthy controls for us than people who are sick, because you understand that we can't do this work unless we compare it to something. So if you have healthy controls that would like to help us understand the difference between healthy and sick, we're recruiting several studies that need healthy controls, and particularly women healthy controls, men too. This study is already recruiting. This is a microbiome study where we're redirecting the gut to a healthier microbiome, one that doesn't interact with the brain in a negative way. And so it's a kind of easy way to explain the Kanika study. This is looking at people with ME/CFS with and without IBS who are currently not on probiotics. This is a real easy study to be in. It's virtual. We just need a tube of blood at the beginning and a couple of tubes of blood at the beginning, couple tubes of blood at the end, but otherwise the rest of it's on your computer and taking either the probiotic or the placebo for just 1 month. It's a really simple study, but we're sending out a lot of recruitment call outs right now. So if you're interested in the study, we'd like to just get it done, because we get to move on to do even more exciting stuff right after that. So if you could help us with that, think about volunteering for that study. I'm going to highlight a couple of studies. We had a huge COVID pause, like everybody else in research, and it's really particularly desperate in our field where we're just on the almost there set of moving our stuff from bench to clinical care. We were just about to launch this study when we got stopped by the pandemic. And you might know that all research was stopped by the pandemic, with rare exception, because we were so afraid we would hurt people by giving them, or even bringing them in, you know, to bring you into a healthcare setting during the beginning part of this pandemic before there were vaccines. It was just not a good idea. So all the IRBs, which are our review board panels that make this safe for humans, said stop. So we stopped everything that involved human contact, and we just had to revert to our virtual based studies for a while. But when the vaccines came out, and now that they've been proven to be reasonably effective and the variants have shifted a bit to be slightly less deadly, I'm going to say that because there's 248,000 people in ICUs in the United States today with COVID. So don't think this is over. But we have adjusted ourselves to a new reality, which is how to do research as safely as possible. And we're starting again. So that gets us back to this study. This is the exciting reboot study that I've talked about to you over the years. Like, we're going to get there, 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 we're there. I finally found some donors that were willing to support this because we didn't have federal money to help us. And we were just about to start. I'd hired all the staff, we'd gotten stuff ready, and boom, we got stopped. So we have been restarted. We're allowed to go now. We have one more IRB hoop to jump through. So that'll be July. So I'm anticipating the end of August for the start of our reboot studies. So what was this reboot study? This is our moonshot. This is our go for the cure study. We couldn't be more excited about a study like this, because here we used all the data we collected from you guys who were willing to come in and get on a bike and let us draw bloods 9 times over 24 hours, measure every gene that got turned on and off, every cytokine, every hormone, every cell, whether it was activated, what its function was. We did absolutely everything we could think to do 9 times over 24 hours, and then gave that to the systems biologists to put into their supercomputers. Think Watson. Have the supercomputers and there's brilliant computational scientists develop models of what this illness is, why does it persist, what is a relapse, and most importantly, as we've been doing so much research in this field, and we know there's an endocrine problem, an autonomic problem, and an immune system problem, to try to put the horse before the cart. What happens first, second, third? So we can be intervening at the front end of a relapse instead of just treating the tail end and the symptoms of a relapse. Super cool science. I mean, the most exciting thing I've ever been involved in. This is some very early stuff to give you a flavor of that very early. This is at 15 minutes after you put someone on a bike. The top 10 things going on in the immune system, in the gene signaling space, are immune. So immune's really, really early. But actually, now we're learning from even measuring things before 15 minutes. Amazingly, we're measuring things at 3 minutes, 5 minutes, really fast. That autonomic matters too, really, really early. But then at the end, this is 4 hours, not the end, but 4 hours after people are starting to feel like they exercise, now they're going to start lay into a post-exertional relapse. These are the signaling. There's not too much immune system here. This is autonomic and pain and oxidative stress and so on. So that taught us something, that we could use this kind of method to really understand first, second, third, fourth, and then envision interventions that could perhaps stop that. So this is what the computational group did. They constructed the models from what we know about how things are supposed to act, plus what we know about is how it's happening in our experimental designs. Then do these models in the computer that model the entire illness, and proceed to do clinical trials in the computer, in silico clinical trials, hundreds of thousands of studies that push this button only, this button plus this button in different orders at different levels and so on, but just hundreds and hundreds of thousands of potential treatments that are done in silico to identify and optimize the sequence and the delivery of treatments. So basically, we take what we know from our challenge data, we take what we know from biology and the literature, we merge all that knowledge, create models of illness, use virtual clinical trials to try to move people from a sick chronic state to a well chronic state, and then take that back to the lab. If we have an animal model, test it in the animal, and then to humans. So to give you an update on what we were doing there in Gulf War illness, where we have a lot more money, and we also have an animal model, we were able to prove that the model that the computer predicted cured the animal model. Cured, didn't make it better, cured it. And these are animals that had the equivalent of 10 years of illness. So very exciting to us that the computer could predict for us a potential treatment that was potentially curative in an animal model. We were funded to move forward to humans. We just completed our first phase 1 study, and we have a second phase 1 study to tweak the treatment. We feel like we're on it. We moved the needle. We didn't move it quite where we wanted, but almost. And now the second study is trying to push it over all the way. So we're well along in the human studies in Gulf War illness. In ME/CFS, since there is no animal model, but because the animal model is, because the drugs that we're using have known safety profiles, or FDA approved the repurposed drugs, we're moving straight to human, and we're going to hopefully be able to jump over the animal model stage of this and you know, not worry that we lost 2 years to the pandemic in our timeline. This is kind of the cool thing that this kind of method uses. It can look for these targets, and so you can see in ME/CFS that they found 11 targetable gene spots in this modeling system, identify the genes, and then they can go to a specific gene and say, what are all the drugs that touch that gene? It's really interesting. And you know, 11 different times you do that, but then you combine it with all the other knowledge and create this virtual clinical trial space. And then not only identify the target, but identify the treatment, the potential treatment, and then model how we would best do that. In all of our models, we never could fix this illness with a single drug. You can't fix it by targeting just one system, like just doing the immune system. We had to do immune endocrine or endocrine autonomic, or we had to combine systems to be able to find always a 2 or even 3 button approach to be able to propose really definitive types of responses. So this is what the cognitive team does. They find the targets, they optimize the treatment, they minimize adverse drug effects and so on, all by using these computational approaches before they hand it off to the clinical trials team to develop the trials and move it forward. And then they take all the data we create and put it back into the model. So in our clinical trials, we actually put people back on the bike and do the whole 9 million data points per time point, 9 times thing per patient to give these guys back the modeling data they need to prove whether or not their predictions worked and to tweak and improve on these models. So that's our going for the cure. We're going after a multi-system system, and we're on it. I'm going to jump over this because for time. So that's we're doing right now. We have that study, which will be recruiting at the end of August. We have the Kanika study. Hey, you can do that in the meanwhile because it only takes a month. And we're really would be happy if you would reach out to us and sign up for that study. And I had a slide for that. I think I put it in a different spot, so I'll find it for you. I'll put it up in the Q&A session so I don't take up too much time. Okay, so that's part 1 of what I'm supposed to talk about. And now I'm supposed to talk about vaccines. All right, this is a short talk, and how much time do I have left, Sarah?
We're over, but
okay.
I think this is very relevant.
5 minutes, so
Perfect.
real quick. Okay, I'm sorry.
Nope, that's okay. Thank you.
So this is the most common question we get in clinic. Every single patient that walks into clinic asks me this question. And the backdrop of this is people with ME/CFS have hyperreactive immune systems. Many of you have mast cell activation syndrome. Many of you have autoimmune disorders, either known or unknown, behind your illness. And so everyone's worried. That makes sense. And I can do this. I apologize for my technical idiocy. My thing is not, oh, there we go. Okay, so first to say, how do vaccines work? There's 3 basic vaccines out there. The one that most everyone took, the mRNA vaccine, the first one to come out, that was Pfizer and Moderna. The second one, the one that just was approved yesterday, was the one that's an old-fashioned vaccine that uses adjuvant and bits and pieces of virus. And the third one is the J&J vaccine that used a viral vector to expose basically mRNA to your immune system. So we have all these different things, but vaccines work by seeing something that shouldn't be there and reacting to it. And they're designed to try to make really big reactions so that your body will make a long-term memory response to that vaccine. And so mRNA is protein subunits, the one like Novavax, it just got approved, or vector vaccines, which is J&J. So most everybody got the original vaccine, not everybody, and they got it twice. A lot of people asked, should they have it the third time? Because there is a recommendation right away, 1, 2, 3, to get 3 shots if you're very immunocompromised. However, the only people in my population that meet that criteria are the people that are on really significant immunosuppressive drugs. Okay, so most ME/CFS patients do not meet the criteria or need to have a third initial dose. So that's the first question that comes up a lot, and mostly the answer is no. If you were on, say, rituximab or some really big gun immunosuppressant, the answer would be yes. But the answer is no for almost everybody else. So the next question is, should I have a booster? And the answer is yes, but. So there's always a but, because everybody's different. Those of you in my practice that have had vaccines, a lot of you guys told me you just sailed through. You were shocked. You hardly had a problem at all. And other people said, oh my God, the worst thing that ever happened to me was get my COVID vaccine, just like every other vaccine I've ever had. It knocked me off my feet. So that was your clue, that if you had trouble with vaccines in the past, you probably need to be careful in the future. So this is, I put some recommendations up that actually are more official than me, just talking about what I think, but I am an immunologist, I get this stuff. But the Mast Cell Disease Society put up a nice recommendation, and I'm going to say this is probably the best one for ME/CFS, because most ME/CFS patients at some level have mast cell stuff going on, and mast cells are highly inflammatory and they react to vaccines. They're the main reason why vaccines give you grief. And so, so taking care of your mast cells becomes the most important thing you can do and to be safer in going forward. So this is the Mast Cell Society's recommendations. Do get the vaccine. Pay attention to how you responded before. If you were already on your mast cell drugs before and you did well, then for heaven's sakes, keep taking them. It's more or less the common sense of this. But that there are drugs that can make it safer, and I've got a slide for that. But these slides will be available to you, and you can go to these websites and read about it. Now, this is the American College of Rheumatology. They think about autoimmunity, and they're saying, do take vaccines. Even if you have an autoimmune disease and you tend to be hyperinflammatory, it's more important to live through a pandemic than to be completely concerned about a relapse of your autoimmune disease because of a vaccine. Nonetheless, there is a risk of a relapse of your autoimmune disease because of a vaccine. And so there's things you might want to do. If you just can't be vaccinated because of past experiences that were god-awful, then make sure everyone around you is vaccinated. That's called cocooning. That means everybody around you is safer and it makes your exposures less. If you're going to start an immunosuppressive therapy for some reason, get vaccinated before you do, so your immune system memory is cemented before you start screwing it up by taking immunosuppressive therapies. Vaccines have roughly a 10% risk of flaring an autoimmune condition. So it's real, but it can be much lessened by taking pre-medications. So what are those pre-medications? These are them. Most important slide I got here. The mast cell is the main thing that's signaling all this inflammation to your immune system, so you can stabilize mast cells. Now, to stabilize mast cells, you got to stabilize them for a couple of days before you get vaccinated. They don't stabilize quickly. So being on a medication or a nutraceutical that quiets mast cells by stabilizing them is important. Quercetin, which I always mispronounce, luteolin, are 2 herbal drugs out there. I'd recommend that you watch out for luteolin because they're olive oil sources that are very safe. And there's peanut oil sources that, you know, in an allergic population sounds kind of nuts to use peanuts. So watch your source on luteolin. Ketotifen is a antihistamine. It's available in Canada and all of the EU and England, all over the world except the United States, as a prescription antihistamine that's a mast cell stabilizer. In this country, you can have it compounded. You can get it because we have the source material that's approved by the FDA. So you can get it, but you have to get it as a compounded drug, or you have to get it from out of country. Aspirin, used carefully because some people are allergic to aspirin, is also a mast cell stabilizer at some level, very mildly, and vitamin C is also helpful. So we usually recommend quercetin and luteolin because you can get it. It's easy. If you've got ketotifen, that's a good one. These are all additives. The more, they work better together. Now, nonetheless, you're going to leak some. So mopping up what leaked, and that's the histamines. The histamines are just one of the 30 plus mediators that come out of a mast cell when it's being inflammatory, but they're awful important. So H1 plus H2. Antihistamines that you take for colds and flus are called H1 antihistamines. And the ones you take to prevent stomach ulcers are called H2s. So when you use H1 plus H2 together, you get a 10-fold increase in the antihistamine effect, which is pretty cool. So you can take your everyday antihistamine, the one you tolerate, it's Zyrtec or Sertrazine's a common one, Allegro, which is Phenodine, but taking it with an H2 like famotidine, which is Pepcid. And then mopping up the rest of it, a bunch of leukotrienes. There's a Singulair, which is montelukast, a common prescription drug. So these are available to you, and you should consider that. Oops. Try again, Nancy. There we go. Stop it. Hang on one sec while I fix my mistakes. There we go. Just to say that we got bragging rights. We have Dr. Harris Theoharides joining our team July 1st. He's one of the world's foremost mast cell experts, and his colleague that was worked with him years ago and is circling back to work with us and with him again, Dr. Durasamy, who just joined our team on Wednesday. So we have two great big mast cell wizards joining our team. We couldn't be more excited. And if you see Dr. Theoharides' website, Mast Cell Master, you'll learn a lot more about mast cells than you ever knew before. You might want to consider that. That's it. Thank you very much.
Thank you, Dr. Klimas, for that research update and how to safely take vaccines. I'm going to have you on standby because I know you're going to finish up our next presentation, but in the meantime, I would like to introduce our next speaker, Dr. Annette Fornos. Dr. Fornos is board certified in internal medicine and certified in functional medicine by the Institute for Functional Medicine. Dr. Fornos attended the University of Miami's Miller School of Medicine and went on to complete her residency at Jackson Memorial Hospital. Before joining our group, she was an assistant professor at the University of Miami's Miller School of Medicine. And I'm going to now hand it over to you, Dr. Fornos.
So thank you, everyone. My presentation, it's about the management of long COVID symptoms, a summary from the recommendations of the American Academy for Anti-Aging Medicine and the American Institute for Functional Medicine.
Dr. Fornos, can you have it so that it's taking it? Can you like start? Oh, okay. Okay.
Is that okay? All right. Okay. So then disclaimers, this information is intended for educational purposes. I do encourage everyone who would like to apply some of the recommendations to have the consent of their physician or their medical health specialist. Post-COVID syndrome, the COVID-19 pandemic has given rise to a new pandemic called long haulers or post-COVID syndrome. The diagnosis of long haulers phenomenon or post-COVID syndrome has to be made after there is a documented diagnosis of SARS-CoV-2 infection. So this poses difficulty in perhaps not being able to diagnose all patients that suffer from post-COVID syndrome, in that some patients with acute COVID may not suffer enough symptoms to go and take a test and be diagnosed. And some patients just simply do not get tested and diagnosed in general. And in addition to that, there are patients who have pre-existing conditions or symptoms already prior to COVID-19 infection that got aggravated or exacerbated, or new symptoms appear prior to post-COVID-19. And unfortunately, by definition, it's very difficult to attribute it to just the having had the SARS-CoV-2 virus. Post-COVID syndrome, there are 4 stages. The first stage is related to the acute infection. It's a period of about 4 to 5 weeks post-infection, which is called just post-infection related symptoms phase. Then there is an acute post-COVID symptom phase from week 5 through 12, followed by a long COVID phase from weeks 12 through 24, and then the persistent long COVID phase from weeks 24 on. And these are some of the symptoms that are associated most commonly with post-COVID syndrome, fatigue being a major one, pulmonary symptoms such as shortness of breath, cough, wheezing, upper respiratory, rhinorrhea, sore throat. We have muscle and joint involvement with joint pains, myalgias, intermittent fevers. In the lungs, you can also see pulmonary fibrotic changes on imaging studies. Cardiovascular system, palpitations, inflammation of the heart muscle or myocarditis, chest pain, cardiac arrhythmias. We can also see acute kidney injury, dermatologic manifestations, hair loss, neurologic manifestations, including loss or decreased sense of smell and taste, POTS or autonomic dysfunction, syncope or pre-syncopal episodes, increased risk for thrombotic events, and so forth. Very importantly to notice is the effect on the GI tract. Patients who have contracted SARS-CoV-2 have a dysregulation of the gut microbiota, which goes into why we're doing research in this area. Next slide. I just want to present, the purpose of my presentation is to go over some nutraceuticals, vitamins, and minerals used for the treatment of post-COVID syndrome. There are many more modalities that are not included in my presentation. The first one I want to mention is melatonin. As we know, it's widely used for sleep. It has many functions, including with COVID-19 for protection against viral entry and replication by decreasing the surface expressions of the ACE2 receptor, and also through certain enzymes intracellularly that are inhibited or decreasing activity that the virus uses for active replication. It also has antioxidant and free radical scavenging properties. It increases major antioxidant enzymes such as superoxide dismutase and catalase. It increases glutathione. It has anti-inflammatory properties by decreasing the pro-inflammatory cascade, in this case, NF-kappa B, toll-like receptor 4 activation, which activates at the cellular level the inflammatory pathway in the production of cytokines. Also TNF-alpha, IL-6, and the angiotensin 2 effects. It increases SIR2. Sirtuins are enzymes that they regulate whether a cell is going to go for cellular death or apoptosis, or if we're going to repair the DNA damage in that particular cell. So it's sort of like an honor of a surveillance system to protect us in general. And it also decreased the activity of macrophages. Through all these mechanisms, it makes sense that it will reduce the cytokine storm. Melatonin has been, the insufficient amount of melatonin poses a risk for increased susceptibility to infection and complications. And one little something I want to notice is that small children who tend to have very mild infection, if any symptoms, they have much higher level of melatonin than adults. Cautions with melatonin, like with any nutraceutical or medication, there's got to be some caution, especially in patients who suffer from psychiatric conditions or on psychiatric medications, pregnant women, breastfeeding women, etc. Long-term high doses of melatonin have been known to reduce serotonin levels because serotonin is a precursor for melatonin. And it also affects the mechanism of multiple drugs by affecting the cytochrome P450 metabolic pathway in the liver. Vitamin D, there have been multiple studies about the beneficial effects of vitamin D as an immunomodulatory vitamin, modulating both the innate and the adaptive immune systems. It also modulates the renin-angiotensin-aldosterone system in the kidneys and lungs. It also exerts antithrombotic effects on the endothelium, or the innermost layer of blood vessels. Chronic conditions that increase susceptibility to SARS-CoV-2, such as age, advanced stage cancer, immunocompromised stage, elderly patients, especially patients who are nursing home residents, are also found to have deficient vitamin D levels. Low dose naltrexone, we use it widely in the treatment of ME/CFS. Naltrexone at higher doses is a non-selective antagonist of opioid receptors used for acute opioid intoxication treatment and opioid addiction. The off-label use of naltrexone, or low-dose naltrexone, acts as an agonist of the opioid receptor and has anti-inflammatory properties. It helps with decreased autoimmune diseases. It helps with chronic pain, obesity, and cytokine storm. With potential side effects of LDN, the one that I most commonly see is insomnia or vivid dreams, and this is easily solved by just taking it at a different time of day rather than bedtime for the patients who need to do that adjustment. Paradoxically, it may also lead to fatigue, loss of appetite or decrease in appetite, nausea, hair thinning, mood swings, and mild disorientation. What I've seen mostly in my practice is the sleep disruption, which is easily pretty much corrected. And I have seen some mild GI symptoms. And in any case, patients who do not tolerate it at those doses, the doses can be titrated down. We start very slow with our patients and advance we start very low and advance very slowly. And ultimately, patients who don't tolerate it, well, they can safely discontinue. With potential long-term side effects, there is possible liver and kidney toxicity, for which we recommend always monitoring labs in all patients who are on LDN. And there's possible tolerance after use for a long period of time, especially at high doses. The absolute contraindications for LDN, patients who are on opioid substances, due to what I've mentioned before, patients who have active, acute liver failure, acute hepatitis, active liver failure, and patients who have active alcohol and substance abuse disorders. I want to briefly mention the issue with mitochondrial dysfunction. The mitochondria are the intracellular organelles that classically produce energy for all metabolic functions and energy for living in general. So it makes sense that any condition that affects mitochondrial health will lead to reduced energy in the form of ATP, increased oxidative damage from excess free radical production. Free radicals are an intermediary path in the production of ATP in the electron transport chain. So any faulty mechanism of this process will lead to excess free radical production, which will lead to alterations in glucose metabolism, pain sensitization, neuroinflammation, and faulty immune defense, among others. And ultimately, it can also pose a risk for cancer. So it makes sense that we need to support our mitochondria. There are many ways to support the mitochondria through nutrition, caloric deficit, fasting. The aim of my study is to focus on the nutraceuticals that we use for mitochondrial support. Mitochondria get the energy from carbs, fats, and proteins as the substrates for energy production that requires cofactors such as B vitamins, lipoic acid, and L-carnitine. I will mention a little bit more about those two later. Then we need entry into the citric acid cycle through acetyl or acetyl-CoA, and these need B5 as a cofactor. In the citric acid cycle, we need other cofactors, again, B vitamins, again, lipoic acid, and glutathione, magnesium, iron, CoQ10, and manganese. Energy transporters such as niacin and riboflavin, and finally the electron transport chain, which requires those cofactors. And again, we see CoQ10, alpha lipoic acid, and magnesium, among others. So improving mitochondrial health, it makes sense that by supplying these cofactors, nutrients for the mitochondria, we are going to heal or help heal the mitochondria or help it just stay healthy. And some of the nutraceuticals, like I mentioned, magnesium in the form of magnesium glycinate or threonate, coenzyme CoQ10, when combined with selenium, it increases the capacity for, to decrease oxidative stress and inflammation. Alpha lipoic acid is a very good antioxidant and anti-inflammatory. It decreases inflammatory cytokines such as IL-6 and TNF-beta. It decreases CRP, so reactive protein, protects the endothelium by restoring nitric oxide, enhances the reduced glutathione available for antioxidant capability. And with ALA, it has to be monitored by your healthcare provider because it has the potential to decrease the conversion of T4 to T3 by the thyroid gland, and therefore thyroid function testing needs to be done periodically. D-ribose is a monosaccharide simple sugar that is essential for the mitochondrial function. NADH, it's also necessary to replenish ATP. It's a strong antioxidant and it's also a free radical scavenger. And then finally, L-carnitine. As I mentioned before, it's an antioxidant, anti-inflammatory. It's used in the shuttle that transports free fatty acids into the Krebs cycle for energy utilization. And it also helps the development of and maturation of T lymphocytes, inhibiting reactive oxygen species production and modulating cytokines. With L-carnitine, it's important to measure if we're going to do L-carnitine at higher doses or long-term. It's important to measure the TMAO or trimethylamine oxide levels because this is produced in the colon by intestinal microbiota. That if it's dysregulated or dysbiotic, it would increase the levels of TMAO, which is in itself a pro-inflammatory, promotes vascular inflammation. B vitamins, besides what I've mentioned above, participate in activating both the innate and the adaptive immune response. They reduce the production of pro-inflammatory cytokines. They protect the endothelium. They reduce hypercoagulability. And for those hospitalized with SARS-CoV-2, it may reduce the hospital stay. Zinc has been utilized widely to enhance the immune system because it boosts the acquired immune response. It also has anti-inflammatory action, and the elderly, it's a population particularly susceptible to both zinc deficiency and SARS-CoV-2 complications and severity. We have quercetin. Quercetin is derived in nature from polyphenols such as onions, berries, and it is a very potent anti-inflammatory antioxidant agent. It has analgesic properties and it has antithrombotic properties as well. It directly inhibits the one of the inflammasomes that is basically a pathway by the cell has to respond to cellular injury, whether viral or cellular debris from damage. It activates nuclear pathways to produce pro-inflammatory cytokines. When acutely done, it's necessary to reduce infection, but when it stays activated for a long time, it leads to chronic inflammation that is unregulated. So quercetin helps to inhibit the inflammasome, in this case, NLRP3. The co-infection of quercetin with vitamin C has synergistic effects, therefore increasing both their efficacy. Glutathione, like I mentioned before, this is a master endogenous or self-made, produced by the body endogenous antioxidant. It has strong antiviral, anti-inflammatory, anticoagulant properties. It's postulated that glutathione or inherent glutathione deficiency it's directly correlated with the severity of the SARS-CoV-2 complications. I mentioned probiotics. Dr. Klimas mentioned probiotics before, and so the infection with SARS-CoV-2 will lead to dysregulation of the gut microbiota, which will increase inflammation. Immunity begins in the GI tract. So I want to skip this slide for a second and just to mention that probiotics, not only to increase our gut microbiota diversity, but also specific probiotics may have specific effects. For instance, the probiotic Saccharomyces boulardii, which is a helpful, healthy yeast, blocks NF-kappa-B and TNF-alpha, TNF-gamma, IL, interleukin-8. So this is a summary of the nutraceuticals that I have mentioned and covered. And I just want to stress out that these aren't the only ones. There are multiple nutraceuticals, there are multiple approaches, and these are just one of the most commonly ones studied. And this is an example of additional ones such as resveratrol and green tea. Green tea is an excellent antioxidant, and also don't forget your diet rich in phytonutrients and healthy fats, omegas, healthy sources of omega-3 fatty acids, DHA and EPA, and from the omegas come really important pro-resolving mediators whose action is to actually decrease that chronic inflammatory state that's common to post-COVID syndrome and other chronic inflammatory illnesses. And this is, again, I'm going over the same supplements I mentioned. In addition, you can see curcumin is also there, antioxidant, anti-inflammatory, MAC and acetylcysteine as a precursor for liposomal glutathione, omega-3s, like I mentioned before, and all the other ones that I just covered. And I think this concludes what I had prepared, and I thank you for your attention. I'm going to pass it on again to Sarah.
Thank you, Dr. Klimas. I know you have a second half to that for long COVID research.
Yep, hang on, I am going to share screen. So I separated long COVID from ME/CFS because that other slide, had 14 different ME/CFS studies that we're doing or recently completed or whatever. So long COVID's a bit of a different story. It's newer, of course, it's harder to get funding real quick, but we are very excited to be the COVID Up site. I'll show a slide on that. That's a CDC funded study that is looking at the comparison of long COVID to ME/CFS. Now, that is a big deal because the Recover COVID network is not doing that. And that's a shame, but we are doing that. And it's a study that's underway, and I'll talk a little bit more about it. We have submitted to the NIH a couple of studies. One of them is a clinical trial with the people that are trying to develop Inspiritol, which is a really cool potential intervention for both ME/CFS and long COVID. We've put our foot down. I hope that I can stay firm on this, that we're not doing clinical trials that don't have an ME/CFS comparator group. And honestly, I think ME/CFS is, we all hope long COVID answers the questions that needed to be answered for ME/CFS, but if you don't compare them, how are you going to know? And that is the bottom line. The scientific question is not being asked. And so when the $1.15 billion has been put into post-COVID illness by Congress, and that's a wonderful, is spent, if at the end of the day we can't say that we know whether or not it applies to ME/CFS, then the ME/CFS patients were left in the dust one more time. So I'm trying very, very hard to make sure that we do this right, and that whenever we can negotiate it, we will be putting in the comparator group. So in Inspiritol, we did that, which is wonderful, and they were totally open. That's what they wanted to do. We're also meeting with people that have a stem cell intervention actually next Friday, and they've agreed to put the comparator group in. That's very exciting. We also have a study that's sort of on a thread right now, but I hope to be able to reel it in, that's specifically looking at people with long COVID that still have persistent COVID, which is a subgroup of long COVID. And there's some potential there that the treatments that are specific for COVID could be very helpful. So we're trying to get that one. I can't promise I will, but I am trying. So that's what we're doing. I do want to take my bully pulpit for a moment to say that after 30 years personally, plus of doing, how many years has it been? More than that, 40 for me. It has been 45 years of doing ME/CFS research, for better or for worse. That's how I spent my career. So, so a long, long time. And what I learned from that is applicable to the long COVID people. It is way better to be a splitter than a lumper. That means that when you're doing clinical trials and you're focusing down on something, like I just said, what about the people with persistent COVID infection? It's not going to be everybody. It is not everybody, but it is going to be some people, in which case that would be a subgroup with an appropriately focused treatment. That's how you should ought to do this thing. So I'm a big proponent of subgrouping, biologically subgrouping people and putting targeted treatments at the right subgroup. So lumping versus splitting, I'm a splitter. And I think that's an important lesson learned from the ME/CFS world that needs to be taken to heart to the post-COVID world early. So you don't waste a lot of time doing clinical trials that fail because you had the wrong entry criteria. That's really, really important. I don't know how to underscore that. So I'm going to spend a minute talking about our COVID UP study. COVID UP stands for Understanding the Post-Viral Phase, COVID UP. It's a great study. We're so excited about this study. I'm doing this with Dr. Anna Palacio as the co-principal investigator, Dr. Hardigan here, Christina Enley, who's on the call right now. I suspect Irina and Violetta, you know, and Allison and the rest of our team. We're doing this amazing group of people that are trying to do a phenotyping study of post-COVID illness that aligns with the giant phenotyping study we had completed right before the pandemic on ME/CFS. It's great. We have this data set of 750 people that went through the phenotyping study that the CDC did right before this, that was done in 7 clinical practices of very well-known, very well-respected ME/CFS experts. So there's no question that the people we studied had the illness. That's always a bone of contention in a illness that has such subjective entry criteria. So anyway, we have that data set for comparison. So we aligned the studies so that we were studying a lot of the same elements. And then we proceeded to try to recruit this long COVID study. Our long COVID study has the recruitment criteria has it coming through the South Florida healthcare systems to get to us. So Jackson Memorial, Broward Health, Baptist, NSU Health, University of Miami's healthcare system, and so on. All these big healthcare systems have been sending out emails to the people that are COVID positive in their database in the last year, 1 year. That's one of our entry criteria. And then they hit a link and they get in. But I just sent out an email to people who think they fit and didn't think they got the email. So I use the advocacy groups to try to reach out to a larger group. And we have ways to talk to you and make sure that you get into this study if you're eligible for this study. So please follow up on that one. So anyway, COVID UP, we're doing a longitudinal study. That means online, we're measuring things that you go on the computer and you fill out your assessments on severity of illness and what's sick about you and how bad is it. And then we bring in 200 people into the clinic and do all kinds of stuff, echoes and spirometry and a whole lot of biologic work in the laboratory and a really thorough evaluations. So it's a very exciting study. And of course, all that's free to the participant. All that data comes back to you. If it's clinical data, you'll have back for your providers. It's very exciting. And as I say, it's comparing to this, the MCAM study, which we just had completed right before the pandemic. So it's topical and relevant. And I don't think of a better data set to compare to. And this study is published. So if you want to read more about it, you can see what our design was. But there's a lot of really great people in this research group, as you can see some of your big names, Dr. Bateman, Dr. Lapp, Dr. Podell, Dr. Peterson, Dr. Nadelson, just really, amazing people, plus that really amazing team at the CDC, who, by the way, fails to get the respect they deserve. They've really dedicated themselves to trying to understand this illness, and somehow they get slammed with every governmental, thing that you're angry about. But the actual scientists at the CDC are doing a wonderful job, and I really love working with them. So it's like they get no respect, but they're amazing scientists, and I hope that you could be a little compassionate when you're angry at the way life is, and because it's not up to them how much money got spent on this illness. So these are my take-home points. To be able to really benefit as the ME/CFS community from the long COVID investment, we need studies that have the ME/CFS comparator group. Can I say that louder? We need ME/CFS comparator groups in the long COVID studies, or we won't be able to generalize what we learned from long COVID to ME/CFS and apply it and get you guys, everybody, forward. We got to all work together to move this forward. And I know the ME/CFS advocacy community really helped the long COVID community get that kind of investment, really worked hard to help, make this happen. And I really hope that the long COVID community, who has not had to live through decades of frustration the way the ME/CFS community has, can see that they have the opportunity to help their brothers and sisters with this illness by advocating and by being active in asking for that. So the COVID Up study right now is your go-to study for the comparative group. We need to be successful in that study. I need help recruiting that study. And I know it's not a straight up recruitment. It has to go through this side door. But if you think you're in South Florida and you might be eligible, reach out to us. We really need you. If you have long COVID, we really, really, really need you to sign up. We only have, I don't know, about 50 or 60 people in the phenotyping study so far. I need another 140. There's lots of room in this study. So please reach out. And again, at the end of this day, there'll be slides that can show you how to reach us. On the clinical trials, I am saying this is really good news for the ME/CFS community. For the pharmaceutical companies are talking to us. Before they were closing their doors, you couldn't even talk to them. Now they're like, well, tell me about long COVID. They realize there's this huge, I've calculated on 800,000 long COVIDs in South Florida from the data that we know about. That's a lot of people. Nationally, obviously the numbers are huge. What are we at? 60 million known COVID infections. So do the math on that, maybe 1/3 of those. That's a huge population. They're coming, they're knocking at the door, and then they want to hear about ME/CFS. So that's exciting. I'm very happy to report that. That's all I got. Thank you very much.
Thank you, Dr. Klimas. Our next speaker is Dr. Irma Rey. Dr. Rey is a physician at the Institute for Neuro-Immune Medicine and assistant professor for the College of Osteopathic Medicine at Nova Southeastern University. Dr. Rey is board certified in internal medicine and environmental medicine, primarily treating patients with immune deficiencies such as chronic fatigue syndrome. Dr. Rey specializes in mold, environmental toxins, genetic medicine, and the microbiome. I'm going to now turn it over to you, Dr. Rey.
Thank you, Sarah. Okay, so I want to talk about an introduction to an environmental approach to CFS-ME, which also translates to Gulf War illness and long COVID, because this is how I approach all the illnesses that I treat. Anybody that has the level of evidence that I'm presenting is level 2, 3, and 4, which is based on small studies and research on our patients and case reports. No big studies, but I will present something at the end that I think you'll all be pleased by. This is my usual hand diagram that I've been working with for years, and anybody who's been to our conferences has seen this hand diagram a million times. This is the part that I'm addressing. the environmental portion of it, which, as Sarah previously said, you know, I like to look at, you know, whether somebody's been exposed to mold toxins from water damaged buildings or from food, petroleum, pesticides, plastics, electromagnetic forces, Agent Orange, other herbicides like Roundup, and other contaminants that we have in our air and our water supply all over. And, you know, it affects our immune system. It affects our viral status, because it can, you know, if the immune system gets damaged, the chronic viruses can reactivate, and we see a lot of that in our patient population. It, you know, I put down the lab testing that we do for the immune system, among others. We also, I also test for mast cell activation, and it can give you sleep disorders, endocrine disorders, cognitive disorders, and autonomic disorders like POTS, and other forms of autonomic dysfunction. This is the way that the American Academy of Environmental Medicine looks at the way that toxicity affects us. So it's total body toxic load. And many of the things that Dr. Fornos already mentioned, that Dr. Klimas already mentioned. This is Dr. William Rey, who was one of my mentors, and he was one of the founders of the Academy of Environmental Medicine. And he always used to have, he has these barrels all over his books, because it's all the things that are additive, and all those additive chemicals tend to cause various effects in your immune system, in your mitochondrial dysfunction, and your, therefore your energy, and it sets off cytokines which give you other end organ damage in your kidneys, and your brain, and your heart, and your lungs, and in your immune system. So I take a look at all that. This is again another barrel that he had with, this is the part of the brain. I don't know if you can see it. So these are neurologic effects of the total body toxic load, among others. Dr. Rey wrote 7 books. So there's a lot of information. I just wanted to give you a small overview of the things that happen. And they talked about the amygdala retraining that we're going to have. The amygdala is right here as part of this slide. So it all ties in together. It's all part of the things that are affected, of the body systems that are affected in our patients. This is another effect, electromagnetic forces. Right now we're being bombarded by electromagnetic forces from our computer screens. And if somebody is mold toxic or has pesticides, plastics, petroleum, those are all additives. So the electromagnetic forces will actually cause more harmful effects on your system, especially in your central nervous system, if you're exposed to a lot of electromagnetic forces. So my recommendation is to shield your electromagnetic forces, and to not be on them all the time, because they're not good for you if you have any kind of environmental toxicity. This is another book that, you know, I found very helpful, was Dr. Theron Randolph. He was another founder of the Academy of Environmental Medicine, and he explained positive and negative symptoms. So positive symptoms is at the beginning of exposure, you start getting hyper, you might start getting headaches, and then you end up with the negative symptoms after years of exposure and years of end organ damage, where you actually start having the fatigue, you start having the cognitive dysfunction, you start having other symptoms. So all that is well described in this book. This book was written in the 1990s, and way before actually he passed away in 1995, so it was written even before then. But it's amazing how much information I learned from this book, and how much information is available to you. It's written in lay terms so you can understand it, and many of you have a lot of medical knowledge already. So I highly recommend this information. So the environmental approach to chronic fatigue, I try to, it's been documented that exposure to environmental toxins is noted in 20%. This is from Lily Chu's study. And in my population, or the population of patients that I treat, I have more than 20% of my patients that have a exposure to environmental toxins. So you have to take a really thorough environmental history. So I use Dr. Bested's excellent environmental history that includes, your occupation, it includes your dietary habits, it includes any exposure to water damaged building, it includes antibiotics, cigarettes, tobacco, electromagnetic forces, pesticides. If you look at the Environmental Working Group, EWG, there's a website for them that has the tap water for your zip code. These are all volunteer scientists that have gone around measuring water pollution levels all throughout the United States. And it'll tell you for your zip code how many contaminants there are that are within the EPA's standards, but are still harmful chemicals. The travel history also includes the immunization history, as we know from the Gulf War patients, the immunization history is very important there. And the occupational and hobby exposure is very important, because if you're a scientist and you spent a lot of time around benzene, or you spent a lot of time around formaldehyde, or you spent a lot of time around any neurochemical or neurotoxin, that's going to be very important to your having developed chronic fatigue syndrome. Also your hobbies, because if you work with clay, if you work with paints, if you're an artist, and you work with acrylics, you may be exposing yourself to chemicals. And there's just things that you need to know about so that you can try to prevent it with air purification, or wearing gloves, or wearing masks, or doing something to try to mitigate your exposure. And the chemical sensitivity history is very important, because it tells you if somebody's sensitive to a chemical, it means they're probably carrying a lot of that chemical or similar chemicals in their system. There are multiple tests for determining toxic exposures. I don't have any, kind of relationship with any of them, but I've used, many of these tests. The real-time labs, urine for mycotoxin, it's covered by Medicare. The Great Lakes Plains lab is covered by some insurances, not too many here in South Florida. And the Great Plains lab for non-metal toxins that I know of is not covered by any insurance, but they also have a marker of mitochondrial toxicity in it, and it tests for all these different chemical toxins. And then we try to detox you from all those things. Great Plains also tests for glyphosate, which is Roundup. They also have fecal hair, red blood cells, urine, and whole blood testing, the organic acid test, which tells you whether you have any kind of propensity towards Candida or Clostridium, because you're making organic acids that are associated with those infections. And Genova stool and GI effects also will tell you about your, gut flora and your enzymes in your gut. And I listed all the different things that Genova does. Microgen also does PCR testing for different body fluids, so you can find out if you have interstitial cystitis. The other labs may not be picking up the bacteria because the testing may not be sensitive enough to pick it up. I use Quest Labs and LabCorp also to test for blood and urine for heavy metals, and Doctors Data also does that. And last but not least, for patients who have like recurrent sinus infections and upper respiratory infections, nasal, postnasal drip, nasal drainage, they may have biofilm associated with MARCoNS, which is multiple antibiotic resistant organisms. And mold toxin patients are commonly found to have this. So this is a test that's available also. It's not covered by insurance, unfortunately, but it's another tool to try to test and to try to make people feel better. There are multiple resources on whole food chelation of heavy metals. I like to go with whole food chelation of heavy metals because it's not only good for chelation of heavy metals, but it's also good for your microbiome. These are listed in online, and some heavy metals require more chelation. So then I refer to nephrologists or other practitioners that are familiar with heavy metal chelation, such as Dr. Lamas, who did the NIH-funded TACT1 and TACT2 trials, actually treating and reversing coronary artery disease and vascular disease by chelation, because he found heavy metals in patients who had limbs that were about ready to be amputated. Genova stool testing tells you, what bacteria, what yeast, what parasites may be there, and they also have charts that tell you whether you're at risk for inflammatory bowel disease, and therefore, if you haven't had the workup by your gastroenterologist, you have some information to take to the gastroenterologist. And like I mentioned before, biofilm will tell you if you have MRSA or other multiple drug-resistant organisms that need to be treated with BEG spray, which is Bactroban, mupirocin, EDTA to break down the biofilm, and gentamicin to kill the bugs also, or just Bactroban, or even silver. Argentin silver can also work for this. This is off-label, off-label use. So here's the study that I've been doing for the past 5 years, and many of you may have participated in this study. It's 236 patients that we tested urine for mold toxicity, and we found that the most common mold toxin that we found was ochre toxin, which comes from water damaged buildings, and we also found aflatoxin, gliotoxin, and to a lesser degree, the trichothecenes, which are the ones that come from Stachybotrys. So these were all patients that were being treated by us, and the testing was dependent on the medical insurance coverage, because like I said, real-time is covered by Medicare, and Great Plains was used for the other ones. The patients with were, this was a self-funded study. We did not receive any research grants for this, and we compiled this data, and the average prevalence was 91%, and the average prevalence in women was 91%, and men was 93%. We have many less men patients than we have women patients, so therefore the statistics may be skewed a little bit because of that, or they may have a greater exposure because of the different risk factors. I didn't go into the slight difference because it wasn't statistically significant. So the prevalence by gender was, the same. Ochre toxin was the leader, followed by gliotoxin, and then followed by aflatoxin. Aflatoxin is more common in food insecurity. So in food insecure countries, or if food is allowed to spoil before it's sold, or after somebody takes it home because they don't notice that the food has mold on it, you can get aflatoxin from it. And now this is the final result, and this is all through your hard work. We were published February of this year in the International Journal of Environmental Research and Public Health, and we're published online February the 12th. And this is the title of the article: Prevalence of Aspergillus-derived mycotoxins and the distribution in the urinalysis of ME/CFS patients. I have all these medical students and pre-med student, and now she's a resident. She started working with me on the research since she was a first-year student rotating with us. they were tremendously helpful in getting this article published. The academic editors and were also tremendously helpful. And so was Beth Gilbert. She reviewed the article before it was sent for publication multiple times and helped us tremendously. And, this is something that shows that we have a 93% prevalence of mycotoxin in our male patients and the 91% of prevalence in our female patients that we studied. So this is the abstract of the article. The full article is available on PubMed. You can research it. I gave you the links so you can get it from PubMed. And we're already getting invitations to do research with other groups because of this article. So I thank you very much for anyone that participated in this study, and I couldn't do it without you. And thank you very much for listening. That's really all I have. The rest of it is just my references, and they'll be available online. So thank you.
Thank you, Dr. Rey. I see Dr. Klimas is cheering for you, clapping you on. Oh, and also thank you for your presentation on environmental illness and ME/CFS and for sharing your most published work. Our next two speakers I would like to introduce are the Institute's nurse practitioners, Dr. Violetta Renesca and Dr. Irina Rozenfeld. Dr. Rozenfeld is a board-certified nurse practitioner committed to the health of her patients. She obtained a second master's degree in integrative medicine at George Washington University and earned her doctorate in nursing practice from the University of North Florida. Dr. Rozenfeld's focus at the INIM includes ME/CFS, chronic infections, vector-borne illnesses, metabolic syndrome, chronic pain, environmental issues, detoxification, and autoimmune diseases. Dr. Violetta Renesca is a board-certified adult nurse practitioner focusing on functional and integrative approaches to treat patients with complex neuroinflammatory conditions. She works with patients to create personalized treatment plans that addresses root causes of chronic illness. I'm going to now hand it over to you both, Dr. Rozenfeld and Dr. Renesca.
Hi, good afternoon, everyone. Thank you for coming for our early webinar. And we share with Dr. Renesca this talk. We're gonna talk about microbiome. And my talk will be very short and more introductory. I'm gonna talk about disease process. I want you to concentrate and listen Dr. Renesca talk, which will be very helpful for you to treat your symptoms and explain why we're talking about chronic fatigue syndrome and we're talking about microbiome. Oh, sorry. We'll talk about ME/CFS, but in relation to another three organ system, gut, brain, and immune system. So starting with the gut, which is it consists from hollow muscular tube, which start in oral cavity and go through the body all the way to the rectum. There is various accessory organs like liver, pancreas, which were helping assist with digestion by secreting enzymes, which help us to break down the food in the particular components, which can be absorbed by us and actually will be appropriate for absorption by microbiome. And microbiome is it's human body contains a lot of microbiome inside the gut and in the skin and in other places, but we're talking about gut at this point. It's a big amount of bacteria, fungi, viruses. About 0.3% of our body weight composed of bacteria. We are just a big host, and we keeping this a lot of microorganisms inside our digestive tract, which is really important. And this microbiome, which we call it microbiome, microbiota, it interacts with us. It helps us. Sometimes it's a pathogenic and it's not good, but we're gonna concentrate today on a good interaction with our microbiome. It is dynamic interaction. It's like micro ecosystem, which integrated in our microsystem, and our macro ecosystem depends on this microbiome. The composition and the function of the microbiome in the gut, it's extremely diverse and different in each human being. We all have different composition in our gut, and it influenced by our eating habits, by our environmental exposures. As Dr. Rey mentioned, we have a lot of them. It's affected by sleep, it's affected by stress, and also by genetic factors and medical treatments. For example, microbial colonization occur early when child born. If it's a natural labor or it's a C-section, it will be different exposure and different colonization. The kids which born from C-section would have possibility more often develop allergies and asthma. So this is really important. Through the life, microbiome continues to influence our different lifestyle, and remarkably different microbiome right now identified even between healthy patients. Now I wanna mention three systems. Interaction between brain, gut, and immune system. There is more interaction with another organ system, but this is very important part where we which this interaction contributes to some symptoms in chronic fatigue syndrome and some very frequent comorbidities of chronic fatigue syndrome, like IBS, irritable bowel syndrome, and I'm not gonna, because of time, I will not gonna go in big details, but really important interaction between gut and brain. We call it another brain-gut axis, gut-brain axis there. And the central nervous system is considered immune privileged organ. There is a lot of blood vessels that vascularize brain, and they form blood-brain barrier, which supposedly to be a barrier which not allowed to be passed by pathogen or unnecessary molecules. And it's strictly for particular molecules to be open. However, importantly, and blood-brain barrier protects the brain, but immune cells can pass the blood-brain barrier, particularly activated, like functional immune cell, they can enter central nervous system through blood-brain barrier, and also bacterial products from the gut can also, and some unnecessary metabolites from the gut can pass blood-brain barrier, and that can contribute to development on central nervous system certain diseases, degenerative diseases of some our patient symptoms, very frequent symptoms like brain fog, right? So this axis involves pathways such autonomic and enteric nervous system, endocrine system, and also known hypothalamic pituitary adrenal axis, where immune cells and possible microbiota metabolites moving on to the brain, and we may not necessarily with a good intention. Also, here I'm bringing immune system. So we cover a little bit about gut and the brain, and here's the immune system. This picture is very busy. We have this vagus nerve and all immune cells and interaction starting immune system activated in the gut with cells kind of showing the epithelial cell in the gut. Microbes also doing a good job for us. I wanna kind of pass it to probiotics to say that it's important for us to have really good microbiome, and this is why we prescribe probiotics. The microbe produce neuroactive molecules that directly contribute to the communication between gut and the brain. For example, neurotransmitters such acetylcholine, GABA, serotonin, produced by bacteria belonging to Lactobacillus bifidobacteria, and they influence brain cell physiology, and directly and indirectly. Practically 90% of serotonin required for our mood, for behavioral, for sleep, for several functions in the central nervous system produced in the gastrointestinal tract, in the microbiome. And there is a whole list of the vitamins like tryptophan, serotonin precursor also. It's a bacterial metabolites derived from dietary tryptophan, and then they can control central nervous system inflammation through specific mechanism. It is important, this activation, immune activation, which started in the gut and processed to the brain, that activated microglia, which is a cell responsible for maintenance in the brain, for maintenance and control of neuroinflammation. So there is a lot of research right now documenting these pathways. I will very shortly, I want to mention this cell, which was mentioned by another speaker, with a big blue cell with a huge nucleus inside and a blue granules. It's a mast cell. We like to talk about this a lot. Epithelial layers in the gut and the respiratory tract, but we're talking about gut right now, lined by these mast cells, which can be degranulated from bacteria and viruses, from injury, from stress, from allergy. And when it degranulated, it's caused many different reactions, which divide in the like immediate reaction locally with cytokines and histamine production. And there is systemic long-term responses, which is producing a lot of vascular effect and which produce a lot of long-term immune reaction on connective tissue. And as I said, body systemic reaction. Like here we can, I don't want you read this slide right now, but then when you're gonna have an opportunity to look through PowerPoint, you can see the different reaction of degranulation of mast cells and what with mediators of inflammation spread. And we can control this degranulation of mast cell in the gut, and then we probably can prevent development of all these symptoms. And as a conclusion, before I pass it to Dr. Renesca, there is a new study. In the last 10 years, there is more information and more research documenting significant alteration in human microbiome in patients with ME/CFS, and which we can document and see that this increased gut permeability contribute to the symptoms, to oxidative stress and inflammation, and affect disease pathogenesis. And they can work about, we implement, we actually practicing it, therapies which aimed on a microbiota, and we try, and it helps in our clinical care to alleviate some symptoms. And when we remove some symptoms, it's already some steps to recovery. Thank you. And Dr. Renesca will continue this topic.
And good afternoon, everybody. My name is Violetta Renesca. I'm a nurse practitioner at the Institute for Neuro-Immune Medicine. I'm gonna continue in the topic of gut, and my topic is a gut restoration. So our gut lining is affected by many factors: food antigen, stress, infection, disease, toxin. Some of it you guys heard from Dr. Rozenfeld. Those factors make changes in our gut lining. We may develop dysbiosis, which means that some bacteria or some family of bacteria overgrows. We start losing protective mucus layer. Our cells are not tight together and they start getting little gaps where things that's supposed to stay in the gut start leaking out, and that's what we call leaky gut. When that happens, we have issues with malabsorption, malnutrition, increased uptake of toxin, endotoxin, and food antigen. That in return leads to immune activation, inflammation, and systemic disease. This regulation of the GI system can have profound impact on health. Dr. Mayer, who is a famous researcher in the gut health and association of gut health between systemic disease, in his research book, he published this picture to show how gut health leads to problems with gut, leads to all those different diseases. And the red lines that you see, those are the immune signals that were associated with those diseases. A properly functioning digestive system is critical for good health. Problem in the gut health can not only lead to aches, stomach aches, gas, and bloating. It can lead to systemic disease. In functional medicine, we use this 5R framework for gut restoration. When applying various illnesses can lead to improvement of the symptoms or resolution of the illness. So the 5R steps include remove, replace, reinoculate, repair, and rebalance. First step is to remove. You want to remove things that are not good for you, like stress, toxin, pollutant, pathogens, bacteria, viruses like Coxsackie virus, fungal, yeast, parasites. You want to remove foods that you might be allergic or sensitive to. That usually is done under the supervision of the physician or of the provider, and it can be done through testing, stool testing, food allergy panel testing, or it can be done through elimination diet. And the treatments includes medication, herbs, and diet modification. The research showed that reducing food that can trigger systemic reaction reduces inflammation, lower the allergenic load, and provide gut with the dietary basis to allow restoration. The second step is to replacing. You want to replace things that you are missing. As we age, we naturally start producing less digestive factor. Besides that, our diet, medication, stuff like anti-acid proton pump inhibitor, it can affect how much hydrochloric acid we produce in the stomach. If we had the gallbladder surgery, we lose our bile salts, and also diet. So very often we don't eat enough fiber in the diet. So you want to replace all those factors: pancreatic enzyme, bile salts, and fiber. Fiber is very important for gut health because it supports the transit, so it helps with constipation, but it also helps us to absorb toxins and feeds the good bacteria that lives in our gut. The third step is the reinoculation. You want to reintroduce the good flora, good bacteria. We do that with prebiotics, probiotics, and symbiotics. Prebiotics is everything that the good bacteria eats. It's mostly inulin, fibers, and fructooligosaccharides. Probiotics are a good bacteria that lives in our gut. We replace probiotics in the form of supplements, or it can be also in the form of probiotic rich foods. Mostly it's a fermented food like sauerkraut, pickled cucumbers, yogurt, miso, temp. This is all sorts of prebiotics. Symbiotics combine both prebiotics and probiotics. So they combine everything that your gut flora likes to eat and also the good bacteria. The fourth step is to repair. When we have a leaky gut, we're not going to go anywhere until we repair the epithelial tissue. We want to do that with nutrients, proper nutrients. What are the nutrients that are needed for gut repair? Stuff like glutamine, zinc, vitamin D, vitamin E, vitamin B5, A. You want to support your mucosal secretion and mucosal lining, and that will be phosphatidylcholine, siliperi L, marshmallow root, plantain polysaccharides. We want to support the immune system in a gut with lactoferrin or immunoglobulin, and we also want to reduce inflammation with antioxidant and anti-inflammatories like fish oil, curcumin, or green tea. None of it works if we don't take care of the whole body, not only the body, but also the brain, and that includes stress, lifestyle approaches, sleep, exercise. Dealing with the stress will help improve the gut health. We can do it through the psychotherapy, meditation, yoga, biofeedback, mindful eating, and better choices. Mastification, chewing your food properly is very important. We often in a rush, in a hurry, and we just swallow the food quickly. We should chew one bite about 30 times before we swallow it. So by the time it gets to your stomach, it's already pre-digested or mechanically pre-digested. So it's easier for the gut bacteria and your stomach acids to thoroughly digest it and absorb all the nutrients. So in the theme of the gut health, we have a rare opportunity to work with the Kaneka, company called Kaneka that produces all the ubiquinol. And they created the probiotic, and our institute is conducting the study titled The Use of Direct Probiotic in ME CFS. Not all probiotics are created equal, so you want to make sure that you take the right probiotics. The probiotics that this Kaneka offers, they already have proof of results. As Dr. Rozenfeld said, probiotics are included in a wide range of function in GI tract, from digestion, priming the immune system, to producing the neurotransmitters. Probiotics are like a good cops that are in your gut, and they keep watch over the bad guys. So we have this study, and we're looking for participants, patients age 45 to 70 diagnosed with ME/CFS. Patient can have or doesn't have to have IBS diagnosis. The study, total length of the study is about 6 months. The intervention includes 8 weeks of probiotics. People that are already taking probiotic, they can still participate. However, they have to be willing to wash off of the current probiotic so they can take the study probiotic. If anybody is interested, please contact our study coordinator, Dr. Anthony Park. You have his phone number here, you have his email. I'm gonna leave this for a little bit so you guys can write it down. If somebody doesn't have a chance to write it down, call our clinic and we can definitely contact you or speak to any of our providers. So as all of you or most of you are our patients, you guys know that we pay closely attention to the gut health, and we work on it very diligently with you. Thank you very much. And this is references. Thank you very much.
Thank you, Dr. Renesca and Dr. Rozenfeld. Our final speaker today is Dr. Craig Tanio. Dr. Tanio is the co-founder of Resolere Health, a medical group that focuses on treatment of patients with chronic complex conditions, including ME/CFS and neurodegenerative conditions. Resolere Health is part of 6 centers across the country that will be conducting a randomized clinical trial of the Bredesen protocol for patients with mild cognitive impairment in 2022. Dr. Tanio received his MD from UC San Francisco and did his internal medicine residency, chief resident year, and Robert Wood Johnson Clinical Scholar Fellowship at the University of Pennsylvania. He is on part-time faculty at Johns Hopkins and at the Dr. Kiran C. Patel College of Osteopathic Medicine here at Nova Southeastern University. I'm gonna now hand it over to you, Dr. Tanio.
Can you hear me now?
Yes, we can hear you now.
Okay, so you can hear me, you can see the screen. Great. All right. So thanks for the introduction. What I'm going to talk to today is around brain health issues in ME/CFS. And certainly brain fog is a prominent symptom with ME/CFS when patients don't come to places like INIM. It's often very poorly understood by primary care physicians or emergency room physicians, and both poorly described and measured. But after this 20-minute conversation, I think you all feel comfortable that it can be measured and quantified with appropriate tools, which we'll discuss today. I do think that a comprehensive approach to understanding the root causes of brain health issues can oftentimes unsurface additional opportunities for successful clinical intervention in ME/CFS. And then improvement in symptoms usually happen either from the brain health side by either directly treating the underlying condition versus treatments that can address the brain health issues more directly. So in using objective biomarkers to understand brain health, there are really 4 different categories. You can measure cognitive performance fairly straightforwardly. There's a couple of products on the market, in particular, something called CNS vital signs that allows one to get validated metrics. It's been evidence-based, covered by insurance. It's easy to administer, and it can be used to follow performance. Certainly validated surveys and doing self-assessment can really be used precisely to pick up what parts of the brain are involved. Anatomical imaging can be helpful to a point. I think one of the issues here is that MRIs, the traditional MRIs, do not pick up neuroinflammation well, which is a problem. There is a software product called NeuroQuant, which does volumetrics, that can be a cost-effective option. I think the addition, cash price in South Florida now is less than $300 in terms of using the MRI as a way to much more effectively measure 3D volume of the brain and pick up inflammation. And then finally, there's functional imaging that can measure brain network and brain function, both QEEG, SPECT scans, and functional MRIs. So here's an example of CNS vital signs, and this is a patient that I saw a couple of years ago who had about 3 years of ME/CFS type of symptoms. Some of the root causes ended up being both viruses, mold, and Lyme. And this was her CNS vitals initially on presentation, total neurocognitive index of 21, but really brain fog that did not affect her memory in much of a way, but really did affect a lot of processing speed and frontal lobe areas. After initial treatment, sort of 6 weeks into treatment, you could see actually that probably because of some of the detox, the memory actually got a bit worse, but some of the other functions started to improve. 14 weeks into treatment, there's another measure. And then finally, about a half a year into treatment, kind of get reasonable recovery of most of the cognitive and processing symptoms. In this patient, there was still a fair amount of emotional symptoms that were left as residual, and we'll talk about ways of picking that up. We do also use detailed assessment surveys. We have a 3 or 4 page brain survey that we'll use on all our patients with fatiguing illnesses. Dr. Datis Karazian has put together, I think, a very good survey that is open source and that we use, that allows symptoms to be matched to different parts of the brain. And then that can be triangulated with volumetric MRI. So this is an example of an output from MRI, which can be used to assess atrophy and inflammation. The company NeuroQuant has put together a validated database that is sort of age sex adjusted. And so the outputs here are in percentiles. So the way to read this is that this patient's frontal lobes are in the 2nd percentile for people her age. And then it gets, and then within the frontal lobes, there are different parts of the anatomy. And so this particular patient has a lot of atrophy of both the frontal and the temporal lobes, and then certainly some relative enlargement of the ventral diencephalon and thalamus, which oftentimes, and caudate, which you see in mast cell activation type of syndromes. And this particular patient had had both COVID and babesia as instigators of fatiguing illness. We have started to use at Rensselaer the quantitative EEG as a way of getting rapid measures of functional brain status. I would say that the equivalent of this now is kind of the handheld ultrasounds like Butterfly IQ, which I think have revolutionized physical exam, where we can take a lot of advantages from technology and just really be able to get nearly a bedside reading. So patients can come in, they get a quantitative EEG, and I can see what's happening in the brain at the time of a first assessment of a patient. Here's a sample output of what happens with a quantitative EEG. There is reproduction. Obviously, it's not an MRI, but there is a comparison of brainwave activity measured by different frequencies, and it's compared to a normal database of controls with excellent emotional and cognitive health. And you can see either hyperactivity, hypoactivity, or problems with coherence. And that can be done in 3 dimensions. There's also peer-reviewed measurements of whether there is a traumatic brain injury, which oftentimes is a confounder and a cofactor in issues with brain health. And then there are more of these 2-dimensional representations of brain maps. And again, what the QEEG does allow for is that MRI is really a lagging indicator, takes a lot of changes for you to see things in MRI, but the functional imaging, you can see effects of treatments within days or weeks. Here is an example of a patient who had COVID and loss of smell. And this is the entorhinal cortex. And you can kind of see with the QEEG measure of lower activity in the entorhinal cortex that really did correlate with symptoms. So all of these 4 types of measurements can be used to precisely measure people's brain health. I've worked very closely with a neurologist named Dale Bredesen, who I think has done quite revolutionary work on using precision medicine as a intervention to put early Alzheimer's and mild cognitive impairment into remission. And this is a framework that he has put together to understand the root causes of neurodegeneration. While many patients with ME/CFS do not have their brain condition advanced enough to go into neurodegeneration, yet I do think this is a really helpful framework to think about things. And how Dr. Bredesen looks at this is he looks at things that encourage the brain neurons and dendrites to create more signaling versus biological pathways that decrease synapses formation. And really, there are things that can basically help the brain connections grow and proliferate, and those that reduce those brain connections. And so if we take it one step down, the type of factors that can affect brain health include circulation. Really a key and basic principle for the brain to work, you have to have enough perfusion. You have to have enough oxygen getting there. And there's so many different ways in which that can be disrupted in ME/CFS, ranging from having decreased perfusion, even if there isn't POTS, that's been documented in a number of studies, as well as hypercoagulability that we're starting to see in long COVID, especially. Certainly hormones can absolutely affect how the brain works, ranging from insulin and glucose metabolism to cortisol, thyroid, and sex hormones. Mitochondrial function is another independent factor, as well as sleep and recovery. In terms of factors that can decrease brain health, clearly the inflammation and cytokine storms that happen in ME/CFS can be a direct factor. Underlying pathogens can also directly infect brain tissue. Traumatic brain injury is a cofactor. Certainly the role of toxins and biotoxins in water damaged buildings plays a important role, as well as chemicals and heavy metals. And then we just discussed the microbiome. And so I think about these two factors sort of in balance with each other. And always the question here is, what is the tipping point? And then finally, you can measure brain network function independently, where we've certainly seen a number of cases where people have recovered from their fatiguing illnesses, but are still left with emotional symptoms or cognitive problems after a number of the fatiguing illnesses have gone away. And what we think is happening there is that the networks themselves have gotten into a pattern and have become dysregulated. And then finally, there can be neurotransmitter balance. We don't have the time today to really talk fully about all of the treatment modes, but I do just want to highlight a couple that we've seen make a tremendous difference in brain health, sort of independently, sometimes of the overall treatment plan. And so really paying attention to cerebral blood flow and oxygenation is critical. And making sure that POTS is appropriately treated, hypercoagulable states are addressed if present, and then independently, hyperbaric oxygen is, there's been a few publications showing the improvement in long COVID, and certainly we can see when we look at QEGs, the before and after changes with hyperbaric oxygen are tremendous. Sleep is always critical. I think one of the challenges with sleep is that it's oftentimes difficult to get accurate reporting on that. And so biomonitors like the Oura Ring, I think, have really increased our ability to get a sense of how people are doing with sleep. Certainly hormone support, especially in the early stages, oftentimes there really is a drop in cortisol that can be supported. In terms of reducing inflammation and cytokines, clearly mast cell activation is almost always present up in early stages, some degree of that. And so protocols there are helpful. Removal of toxins, even if somebody hasn't had an issue with toxins before, oftentimes they can be much more sensitive. And so clean air, clean food, clean living, I think is all important. And then we've seen a lot of examples of where people have come in with triggering events that were never connected to traumatic brain injury. They had prior histories of traumatic brain injury 5 or 10 years ago, but those injuries that were hidden certainly got revealed with the onset of the fatiguing illness. And hyperbaric oxygen, nimodipine, neurofeedback are all really good treatments for traumatic brain injury. Finally, we see in the early stages of many of our patients, and I'll show an example of how this looks on the QEG, of having the entire limbic system activated in terms of the amygdala, the parahippocampus, and other regions. Certainly, there are a number of different programs that are available. I don't think there's enough research to say which one is better than the other, but we've certainly found a lot of benefit from starting to use electric vagal nerve stimulators and neurofeedback. So let me give an example or two of how this looks like. Here's an example of a patient who had fatiguing illnesses for about 3 years. This is the before and after with neurofeedback, where there was really a lot of activation across almost all frequencies of the brain waves. The way to read this is that red is usually 4 standard deviations beyond the norm. White is 0, and then kind of the aqua is hypoactivity. And as we go up into the higher frequencies, almost all of this has been cleaned out. Here is an example on QEEG of a patient who had really significant cognitive decline after having neuro-COVID and had some additional instigators. We treated with both high intensity light therapy and hyperbarics. And what you could see here is the cognitive decline was being really driven by a lot of, this is in the 4th Hertz range, so delta waves that is associated with sleep. The patient was awake, but a lot of the hyperactivity was the equivalent of nearly sleepwalking. And essentially after a week of high intensity light therapy and hyperbarics really helped the brain to recover. We find that light therapy can really be helpful to help the mitochondria in the brain kind of jumpstart. It's not usually a permanent treatment, but rather something that can kind of help in an acute phase while other therapies are available to do more permanent change, such as hyperbarics. So in conclusion, brain fog and brain health can be accurately measured with appropriate tools, including symptom surveys, measurements of cognitive performance, as well as functional and anatomic tools. Taking a comprehensive approach to root causes of brain health can reveal other factors that can be assessed in addition to ME/CFS, and that there is a significant toolkit of interventions that are available today that can work with a precision medicine approach. Be happy to answer any questions as we get into question time. Thank you very much.
Thank you, Dr. Tanio, for your presentation on brain health and ME/CFS. And I also want to thank our speakers for presenting their research and clinical expertise. We now want to open up for any questions. And as a reminder to our attendees, you can use the Q&A box within your webinar control panel to submit your questions. I would like to invite our speakers to turn on their cameras and unmute, and Dr. Rozenfeld will kick it off with some of the questions that came in.
Great. Hi, good afternoon again, everybody, and thank you for our great colleagues today. We have an amazing panel. I personally have a lot of questions, and I appreciate Dr. Klimas already volunteering, typing here that you would like to answer some question. There is a question I can answer. Dr. Klimas may ask, is the Kanika study using an existing probiotic or newly developed one? You wanna?
Oh, for me? Yes, this is a new probiotic. It was developed in Spain. It had a study done with irritable bowel syndrome that was very positive. So they're very excited that they have an IBS focused probiotic. And I was talking to them about it and said, lots of our patients have IBS, but wouldn't it be interesting to know, because we're so worried about the microbiome in general affecting immune activation and immune health in ME/CFS, could we do a study where we compare people with ME/CFS who do and who do not have irritable bowel? So we're recruiting both, people who have it, people who don't. The main restriction in this study is that you can't already be on a probiotic, at least not in the last 6 weeks. So obviously, you can't study somebody else's probiotic. That's not very helpful. So if you're interested in this study and you're not on a probiotic, it's only an 8-week study. It's quick, zip, zip, zip. And we just need you to come in for a blood draw, either the Kendall Clinic or the Davy Clinic before and after, so that we can do the microbiome, all the immunology studies. Of course, you can have those results back, so it's nice for you. And but for us, we're really excited to be doing this study.
Thank you, Dr. Klimas. I want to provide some disclaimer. For purpose of practicing prudent medicine, we will not going to be answer question about appropriate dose or starting dose of medication, low dose, high dose. This is all personalized decision between the patient and clinician. So I apologize, but we will be skipping question about particular. There is no such things on which dose we start in particular medication. It's very, very personal decision of each clinician. I think everybody, my colleagues agree with it, right? So, and moving on, I have a question for, I'm sorry. Dr. Fornos, are you available? I don't see you on the screen. I'm gonna be skipping Dr. Fornos' question for now until she shows, and I will leave it for her to answer. Dr. Rey, on your slide, you mentioned use of shields for EMF. How does one shield for MEF, and where is the resource to be found? Where patient can find recommendation about shielding from EMF?
You have information at the website of the American Academy of Environmental Medicine. You can purchase shields online. You know, I have my phone shielded. There's also rudimentary shields that people create with aluminum. I don't know if anybody saw Better Call Saul, that Netflix series. The brother had electromagnetic illness, and he used aluminum everywhere. And some people do very well with shielding their computers with aluminum and shielding their windows with aluminum if they have EMF meters that are bothering them. And they can also use their shutters. Anything with aluminum will shield, but there are many resources online for shielding. And Dr. Stephanie McCarter, who's an expert in electromagnetic forces, she has lectures online that you can view for that.
What about copper? Is it better than aluminum, the copper?
You got me on that one. I don't know the answer.
All right. And one more question. It's a lot of questions for you. That inner ochratoxin, the OTA abbreviation, I suggest it's a ochratoxin. It's come from foods, and how many people have it in general population also have ochratoxin from food?
Well, there was a study done many years ago on mold toxins, and they studied the general population. It was a very small study, and it was only in the 40% range or something like that. I believe that many of our patients are poor detoxifiers, and that's why they retain the mold toxin in their system, because not everybody retains it in the system. Ochratoxin can come from food, but it mostly comes now from water damaged buildings. There are multiple articles about water damaged buildings and ochratoxin from aspergillus, because the aspergillus / penicillium produces ochratoxin.
Thank you. Oh, so sorry. Dr. Renesca, there is a question for you.
Irina, I'm available if you need me.
Oh,
Sorry,
thank you.
I
I'm
couldn't
going
get
to
it.
give you questions. Thank you. Dr. Renesca, are the same factors what cause leaky gut responsible for compromised blood-brain barrier? I can take this question. And is the 5R approach also the best way to treat leaky brain?
Yes and no. So yes, because leaky gut can lead to alternation in blood-brain barrier, mostly through the inflammation. Leaky gut causes activation of the immune system and increased systemic inflammation. When you have systemic inflammation, you have brain inflammation in general, and brain inflammation and also hypoxia increases blood-brain barrier permeability. So that's why the hyperbaric oxygen can be very helpful, because eliminate one of those factors. So 5R program will not cure the leaky blood-brain barrier. However, if you don't address the gut issues, you're not going to get anywhere. That's why we often start with healing the gut and dealing with the gut, because as Dr. Rozenfeld showed in her presentation, there's a connection between brain and gut. So the body is a complex system that interacts with each other. All the systems talk with each other. So you cannot just treat one little thing, especially in the complex diseases like ME/CFS.
Thank you.
Do you want me to add the leaky brain? Anything?
Yes, Dr.
Go ahead, Dr.
Tanio.
Tanio.
I think on the leaky brain, I think one of the issues with the blood-brain barrier is, what's the practical way to measure whether this is the case? And probably right now, this point in time, measuring sort of S100B and antibodies to that is probably the most available. It's available through Mayo Clinic. Cyrix has a slightly different methodology, but probably not as validated as S100B. Certainly some of the things that help the gut can help heal the brain, but sort of polyphenols, some of the curcuminoids, and then certainly hyperbaric oxygen can also be very helpful in healing the blood-brain barrier.
Dr. Tanio, I have one question for you also. What are your thoughts about using Doppler ultrasound to measure transcranial blood flow?
Yeah, I've not used that as an independent measure. I think with some of the things as we go into brain health, we certainly can go significantly deeper. So for example, I do work with a neuropsychologist, and he'll oftentimes sort of do more extensive neuropsychology, 2 to 3 hour formal evaluations with all the testing, but I don't think that's always readily practical with everyone. And on the transcranial Doppler, I just have not used it. It may be complementary to some of the things, and I'll certainly take a look after this to see if it's a useful addition on top of everything, but haven't used it today.
I have many questions for you. Don't go anywhere yet. Dr. Tanio, I see you did not include neuropsych evaluation as a way to measure brain fog. I appreciate your clarification as to why.
Yeah, and so certainly we will measure. We will do formal neuropsych evaluations when it's indicated, but oftentimes it's a lot for patients to go through. So we may just do a subset that we'll do sort of independently there. But the neuropsych evaluations do have quite the benefit of having significant amount of evidence, being able to be used to sort of track improvement over time, but does have the downside that patients do find it pretty exhausting to go through. So.
One participant suggested, what would you expect to see on EEG of patient with ME/CFS? Would it look like a patient who is ready to fall asleep?
Well, so on that, I'm not sure I have a prototypical, because I do think as we think about ME/CFS, we often, it gets into this question about, multiple, root causes, multiple different types, and really kind of getting into, as Nancy said up front, being a lumper or a splitter. I'm absolutely on the splitter side, and to be as precise as possible. What we will see though is that I do think, let's just kind of talk about, a lot of the patients we're seeing now with COVID that's tipping people over. You do see a lot of involvement of the frontal lobe, tracking, where the olfactory nerve is, enters, and certainly a lot of frontal lobe and executive dysfunction. And so you will see, higher amounts of delta and theta waves sort of emanating from that. Certainly the limbic system is a big issue, and seeing higher activity, oftentimes in higher frequencies in the amygdala and the hippocampus and the parahippocampus is another thing. And you can see that those areas are oftentimes enlarged. Certainly, getting a fatiguing illness can unsurface prior emotional trauma that was, had either been sort of hidden or not fully resolved, and where the emotional history and the actual sort of physical inflammation and changes in neurotransmitters in the brain all come and work together. Hard to say which things precede what, but to know that limbic system is a huge issue, and measuring it, I think, can really help us understand whether certain protocols can work more effectively. And so I've had a number of patients in which some of the commercially available brain retraining programs like DNRS or the Gupta program, they found them transforming. And then we did an EEG and found that there was still a fair amount of hyperactivity, and they were able to use neurofeedback to kind of get that next improvement. But regardless, just using an EEG to see where you're at can be tremendously helpful.
Can patient to do neurofeedback therapy from home?
So it varies. Certainly one of the, and probably maybe the only benefit of COVID that I know of has been is that it did really encourage a lot of virtual treatments. And so in the past, neurofeedback was something that most of the time people went to neurofeedback centers and needed to do it sort of in person. But the technology has evolved now where it can be done remotely and effectively. And that makes it, I think, a lot more accessible to many patients, especially those who are too fatigued to travel. And that's a really important characteristic.
Thank you. Dr. Fornos, do you have, as a functional medicine specialist, what would be your recommendation for severe SIBO, what keeps relapsing?
Sorry, Irina. Can you repeat the question? I can't hear
Yes.
you very well.
What are your recommendations for severe SIBO, what keep relapsing?
Well, that's a difficult question to answer, not knowing the clinical picture, right? One has been tried before, what type of SIBO is present, whether it's methane or hydrogen predominant. So it's, it's very difficult to make a specific recommendation with, with that respect, but definitely testing should be important, and basically the use of different modalities to cover for both methane and, which is more resistant to hydrogen producing. In some patients, I use antibiotics and definitely combined with herbals. For instance, I like biocidin products combination with rifaximin and neomycin in some patients. Some patients I do just herbal therapy. It can depend, and then probiotics are super important. I particularly like Pro4R from biocidin and digestive enzymes with HCl if the patient doesn't have any contraindications to HCl. Hyperchlorhydria is one of the risk factors in SIBO and CFO. So going through the 5-hour protocol, like Dr. Renesca mentioned, is also important for anytime we're treating gut health in general. But for specific recommendations, they have to be made in a patient-by-patient individualized case, because obviously everybody's different and it depends on what has been tried and other conditions the patient may have. So I hope this answers the question.
Yeah.
I have one more question for you, Dr. Fornos. Which of medications and supplements you mentioned will you notice be most effective for long COVID patients who also had
That is a very interesting question. I'll tell you. There hasn't been a comparison, let's say, curcumin versus quercetin. But I do really like quercetin, obviously, in patients who can tolerate B vitamins, melatonin, antioxidant support, glutathione. I think patients who, in whom fatigue is a predominant symptom, I'll try LDN as well. And so I think at least I will try those. So a good multi, you can use like K-Pax or Mitochores for mitochondrial health. It will already include most of the B vitamins, A, alpha lipoic acid. It'll have some CoQ10 in it. Ubiquinol, by all means, for mitochondrial health and energy. So I mentioned the B's, the antioxidants, C, melatonin, vitamin D, absolutely as an immune modulator. So patients don't need to try all of the available supplements for specific condition. There are many, many good ones, but in general, I would focus on mitochondrial health, anti-inflammatory effects, and antioxidant support. And together with all the other modalities that were, some of them were mentioned, that I didn't include in my presentation, which are also very important, right? Addressing the hypothalamic pituitary adrenal axis, working on stress reduction, nutrition, diet, sleep, are also very, very important. So post-COVID syndrome is complicated. We're starting to see the effects of it. It's, duration and complications. We'll be seeing more of that in the future. I think personally that it can be reversed the earlier that you treat it, and perhaps even prevented. It depends on our comorbidities and what we have in preventing the onset of COVID. But in general, these are what I use, what I take, and what I have been using in my patients.
Thank you. I have a question for Dr. Klimas. I don't know what happened with my echo.
What? I
Dr.
can hear you,
Klimas,
Irina. Go ahead.
follow up to PASC with ME/CFS comparator trials question. Interested in EBV in Salmonella. It's a really hard question. I don't understand what.
Yeah, I actually asked to answer that one because of that EBV comment. I'm not sure about Salmonella. I'm not going to be able to answer that. But we, as clinicians, are seeing viral reactivation very commonly in long COVID. There's some papers now showing that people who reactivate EBV while they have acute COVID are more likely to go to long COVID. So it could be that this weird thing that when you hit a virus with another virus, so true in like HIV, that's very true. If you get EBV and HIV at the same time, you're going to have a much worse course of HIV. So, so it could be that if people reactivate their EBV at the time they get their COVID, that they're actually triggering or setting themselves up for the long COVID. We won't know that. Now, EBV is such an interesting virus because it can turn on everything. There's another bunch of questions about polyclonal gammopathies. EBV is one of the viruses that can turn on all of your immune responses to random weird things. Like you could get syphilis antibody and you never even saw the bug, right? It's just one of those viruses that turns everything on. So, and because it's one of those things, it can trigger autoimmunity. So EBV matters a lot to ME/CFS as well as to long COVID. And we are super anxious to do that work. We are trying to talk with some private donors right now, hoping that we'll be able to better understand the role of EBV and if perhaps that's another targetable point in treatment. I'm also going to say my personal bias. If we had an EBV vaccine, we wouldn't have near the ME/CFS or near the long COVID that we have right now. If we had had a vaccine for a virus that with this vaccine technology that's out now, that we should have a vaccine soon, but we don't really know of what's in the works. I don't hear a lot of talk about EBV vaccines coming down the pike. So it wouldn't be nice if we could save a whole bunch of next generations, all the misery that you all have seen.
Thank you. I have an announcement for everyone while we online. If you go to chat and scroll for Nancy Klimas' post, there is a post about Zachary Barnes. He is he's conducting recruitment for all studies in Miami VA. And there is his email, which really easy to remember, zb84@nova.edu. There is a phone number there, and please make a note. You can call Zachary directly and find out about all currently enrolling studies in VA.
I also included information for Anthony Parks for the people that are interested in probiotic study.
You don't have to be a veteran to be in a VA-based study.
Yes.
So the VA is looking for healthy controls. So we're looking for ME/CFS. Don't have to be a veteran. That's that cool study by Dr. Chatterjee that's going to do all the microbiome of the gut and try to connect it to what's going on in the brain. It's a very cool study. So there's a lot that we're doing, and the VA is one of our main partners.
We have a comment from our participant and reply to Dr. Klimas. Moderna is working on EBV vaccine. Thank you so much for
Irina, you want me to
Oh,
answer
good to know.
our question?
yes. I
Okay.
need to go. Hold on.
Someone asked a question about with MRIs, like why patients will feel better sometimes after an MRI. And that's a great question, because I think with MRIs, sometimes you can have people, some people will feel better and some people will feel a lot worse, you know? And so certainly with MRIs of patients with mast cell activation or environmental sensitivity may not be able to handle an MRI, or those who have a lot of limbic system issues, the clicking and the noise is just too much stimulation. But with MRIs, we certainly know with a technology now called transcranial magnetic stimulation that it's actually changing some of the polarization and repolarization of the neurons, where it can be absolute. There's good evidence that it helps in treatment-resistant depression. And so this is, I think, the fact that we can have some people feel better and some people feel worse, just as a good example again of this lumpers versus splitters, that there's a lot going on deeper inside that and there are subsets of patients who will react quite differently to the two therapies.
Thank you. And I want to add, we have a comments about that when you're gonna, for EMF against shielding, copper is, there is a copper nets, there is a copper, you can buy it online through the Academy of Environmental Medicine, Center of Environmental Medicine in Dallas. The copper has a good quality for protection from EMF. And Dr. Klimas, can polyclonal hypergammaglobulin cause adverse COVID-19 vaccines?
So by itself, it's not causing the problem, but it's a signal that you could have a problem, because it's saying, boy, does this person have a hyperactivated immune system. Oh my gosh, it's already flying, right? So then you add a vaccine and you can get even, over that threshold of and get into even more misery. Nonetheless, you have to weigh that vaccine versus risk versus benefit thing personally every time you're dealing with this, and you can't get past the fact that being unvaccinated right now is extremely risky for death. Still, I mean, people seem to think we're past that, but I did mention there's 248,000 Americans in the ICU today with COVID. That's a lot of people, and they're mostly unvaccinated. So, so you have to do this risk versus benefit thing. So I'm just like, well, you know what? The safest way in a real pinch is to use steroids before you take the vaccine. I mentioned that in the typing answers, that we have patients that we just do the mast cell stabilization the usual way. But we have people we really worry about where we go ahead and give them steroids around the vaccine. And we know we will blunt the response to the vaccine, but then we check. And in fact, I've not yet seen someone that we use steroids on that didn't get a very nice vaccine response. So I'm not, I mean, I hear people that won't even take NSAIDs around their vaccine, like a leave-in and that kind of thing. I'm like, seriously, these are powerful vaccines. You're getting great immune responses to them, and they're kind of hard to blunt down. So So I would argue that you do what you need to do for your health, but then double check and make sure you got the result that you were looking for. So checking on levels is a good idea. So you're really in a pinch like that, that's a patient I do steroids on. And I'd pre-medicate for 2 days. It takes a couple days to get the mast cells to quiet down. I'd keep it going for a couple days afterwards, because that's the risky period, the first 48 hours or so after the vaccine, when people are sort of charging on. Also, understand how these vaccines work. People really are confused. You're making mRNA, you're giving an mRNA vaccine that makes a whole bunch of signal so your body makes spike protein, okay? And it does that for roughly 14 days. And then it's gone. It's gone. You can't find any trace of it. It doesn't change your DNA. It's not permanent. All it did was code for something your body made that was the vaccine. So they gave you the stuff that would make your body make the stuff that your body that you were responding to. It's kind of cool. So you don't have a long, you didn't mess with your DNA. You didn't mess with your RNA. You did make a whole lot of virus peptide that wasn't the kind that made you sick. It wasn't a whole virus, just a little piece. And then you made a giant immune response to it. So you're actually being vaccinated for roughly 2 weeks after you take that injection. So your risky period for having inflammatory reactions is roughly 2 weeks long. So even though we're only medicating you for a few days there, because it seems like most people mostly feel their vaccines in a day or two, some people it might drag out, and then we'd have to treat them longer.
Thank you, Dr. Klimas. Dr. Tanio, do you find that brain imaging testing are covered by most insurances, and which are?
So it's with insurance, it's hard to give a one-liner because everybody's policies are different. We are finding that, for example, neuroquants, which were volumetric imaging, about 5 years ago, the coverage was about 0%, and now it's about 40 to 50% or so. So it is varying by insurance, but it's one of those technologies that's been FDA cleared, and it's going up the adoption curve.
So question for anybody who wants to answer, I will try that. What are thoughts on TENS unit connected to the ear to stimulate the vagus nerve? It's not really TENS unit. We have a vagus stimulator. We have some devices which you apply to the neck, the pressure. There is one connected to the ear. I don't think it's a regular TENS unit. It's a little bit more specific device with specific electric currencies. Anybody has comments on it?
Yeah, I think that the vagal stimulators, it's an interesting observation on the US system, where the ones that have been formally FDA approved are surgically implanted for very specific devices and cost about 5 figures or more. And there are some very inexpensive ones that are being adopted more, like in the UK systems and others that have that are less invasive but haven't been formally FDA approved. I think because of the the amount of resources take to get that approval. But we've certainly seen some pretty good results with one that's applied on the ear from Vegas.net, which is over in the UK. And again, I think it's a very promising field. It's hard to tell, I think, at this point, the difference between one electrical stimulator and another. There definitely needs to be more research, but it's I think a very useful therapy. So
I think we almost answered all questions. There is a couple more, just a minute. I think this is a just comments. It's not a question, but it's very promising. What's wrong for me? I'm so sorry. The minute I said it's last question, the question.
I'm sorry, that was me because I was answering it when you were saying it. So she was saying in that question that because she's 65, she thinks it might not be relevant to her. And I was trying to say in my answer that there's research and there's clinical care and there's knowledge, and it's all at different stages. So, you know, for instance, I've been doing this since '87. A lot of people here, God, Irma and I, we've been doing this for a long, long time. The rest of you guys are babies, right? But Craig, you're so young. We've learned a lot as clinicians, and we apply it. And we learn a lot as researchers, and as much as we can clinically apply it. When we do the big research studies, the very traditional way, phase 1, phase 2, phase 3, here this works, ta-da, here's a new clinical guideline that everybody in the whole wide world should be following. That's like classic how it goes, and it does take time. You're right, it usually takes 20 years to go from this spot all the way to that spot. But in the meanwhile, it's not like we're not learning things and we're not applying it in our practice. We absolutely are. What you heard Craig talking about, what you heard Annette, Irina, Violetta, Irma, that's all of us taking knowledge, whether it's from what we've learned ourselves in our practice or what we learn from evidence-based medicine in other fields that we can then apply to our practice because it's been done so well. Then we know what the safety and the potential efficacy is. So I guess what I'm trying to say is there's science and art to being a doctor, and we're not going to see a patient and turn you away and say, I'm waiting for the guidelines. That's ridiculous. So 65, it doesn't matter if you're 65 or 25. When you come in to see a provider, a doctor, a clinician, you're going to get the best we know how to do. And we hope that the best is better with science. And we're working very hard to help with that bigger knowledge. But when you come into the clinic, we're going to just do the best we know how to do. And we'll tell you, you know, how many times have my folks heard me say to a patient, well, let me tell you what I know based on science. Let me tell you what I think based on experience. And we go through it so that you're a partner in this. And when we're trying to make decisions that affect your clinical care, that you know how strong the evidence is and when we're really, really solid in what we're saying versus how we're out there on a limb, but we think it's the right limb, but would you come here with us? Or at least, you know, consider that. And that's how it's got to be. And it's not just in ME/CFS and in long COVID, it's how medicine's actually practiced. We only have perfect medicine guidelines in very few areas, and they're not so perfect. So be glad when you have people who care a lot and read a lot and try to stay up to date. You want out of your doctors a well-educated, well-informed, engaged, and willing to go the extra mile for you to learn even more if need be. And that would be like the perfect doctor for you. So I think I have a whole team of wonderful, perfect doctors and nurse practitioners and alternative medicine practitioners and others that are all trying their very, very, very best to help you be healthier.
I have
Maybe
a question.
Someday what we did will become evidence-based so well that everyone does it that way. That's the goal.
I have many questions on the same topic. It's historical, I would say, and what's the relation of the original? There is relation anywhere between mold exposure and chronic fatigue syndrome?
I'm sorry, I didn't follow the question I was looking for. Say it again.
From the history of chronic fatigue syndrome, can you trace it to the mold exposure?
Oh, the mold. Yeah, I saw Eric asking that question a lot. Hey, Eric, nice to see you again. You're always in my chat box. I know you're a frequent flyer pushing the mold thing, which is important, you know, and what you heard Irma say is that it's super important. If you read the paper she just published, it was shockingly prevalent in our patient population, these mold toxins and mold exposures. So, and I know, Eric, from your perspective, you're looking at it as the inciting event. We also look at it as a perpetuating event, all right? Because a lot of people clearly got their ME/CFS after, you know, mononucleosis or something, and they're cruising along and a hurricane knocks their roof off and the mold happens, and now they're super sick, you know, because it can take someone who was already in immunologic hyperdrive, and they can be much more sensitive. They could be the canary in their house to the toxins that are in their house, which might not be at a threshold that affects people that aren't already sick. So, so we care a lot about molds. The neat thing about molds is if you can discover them, you can try to remove them. I mean, getting rid of the exposure is the number one thing you want to try to do with molds. And then, of course, work hard to get your detoxification systems to work again so that you can get rid of whatever's left. So it's just a lot of work to do. I'm going to go back to personalized medicine. You heard Craig say it, you heard Irina, Violetta, Irma, Annette. Every single patient walks in has a theme song that sounds familiar, but the story is different. And we really have to dig into the nuances there sometimes to find the things we can help and make a difference in how things happen for them.
I want to read one question. It's not a question, it's our mutual patient. Thank you all so very much for the work what you do. We are patient, capital letters, very much appreciated. Big heart. It was not a question, it was a comment for everyone.
Yeah, and I saw a comment in the Q&A that says that when we answered them, that the other people couldn't see them, only the person that asked the question, which I didn't think was true, but we answered like almost 30 questions in the Q&A, typing them out. So if people haven't been able to see them, Sarah, can you say how they get to see those answers?
Yeah, I'll make something available so that you won't see the person who asked the question, but you'll see the question and the answer. I'll make this available following the event.
Wonderful. Thank you so much, Sarah. I appreciate that.
I have a couple answers for, first of all, yes, Institute for Neuro-Immune Medicine accept new patients. We accept new patients. Usually initial visit needs to be done in person, and you need to be seen here in Florida at least once a year for us to establish care with you. We do have telehealth. For now, we do, but each particular state has different requirements, how much we can do over telehealth. When you're gonna be calling our office, make an appointment, our office manager will consult you depending on which state you are, what kind of arrangements can be done for you. Somebody plan to travel for initial visits to see us in Florida. Follow-up visits may be arranged via telehealth, or it can be three-way with your primary care provider and us three together on a call. It's again, depending where do you live. In the state of California and Illinois doesn't allow telehealth visits. It's each state different regulation. But again, we do accept new patients.
Which is different. That's because we have more providers. As you can see, we've gone from just a few to I think 8 right now, which is great. We try to expand the clinic to accommodate. We're very keen to try to help long COVID patients too, because it's my strong belief, I think we all agree with this, that the earlier you intervene, the more likely you'll make a difference. And so I know long COVID patients don't want to get into the longest COVID situation. So if there's a way to accommodate, we're trying to accommodate. So, but I'm going to shout out to you, long COVID patients who maybe don't live in our area. You heard from Annette that the integrative and functional medicine people have a lot on the ball to help with. And there's a lot of integrative medicine and functional medicine doctors out there. So if you can't find someone that can care for you in the way you've been going, whatever that might be, you might look in your network of places and look for a provider that's looking at the whole body and all its regulations and all of its systems, instead of picking apart system by system and having 17 doctors.
And I think we answer all questions. Thank you so much.
I'll take it from here, Irina. Thank you.
Thank you.
Thank you, Irina.
Lastly, I would like to wrap up this event by asking our attendees, if they can, to please consider donating to the INIM by texting NSU INIM to 41444. Your gift, no matter the amount, will help ensure that the INIM will continue to be a vital resource for our community. And feel free to share that text to give code with your family and friends. In the coming days, as I mentioned quite a few times throughout this conference, you will receive a follow-up event email with a link to today's recording, which can also be found on the Institute's YouTube channel. And please don't forget to connect with us on Facebook, Twitter, YouTube, and on our website at nova.edu/NIM. You can also subscribe to the INIM by clicking on the subscribe button on our homepage to stay current on the Institute's upcoming research, events, quarterly newsletters, and much more. On behalf of the Institute for Neuro-Immune Medicine, I want to thank you again for attending. Be safe and have a wonderful day. Thank you.